Okay, I am giving the first talk, Life History, Evolution, and Trade-offs in the Bromeliaceae. And I am a professor at Colorado College, which is an undergraduate serving liberal arts college in the United States. So for those of us lucky enough to be working on Bromeliaceae, I'm here to argue to you today that we're working on a model clade for understanding life history evolution. That is because there's so much variation within our family, particularly between closely related species. What is life history? Well, life history studies are a major branch of evolutionary ecology, which focus on how evolution shapes both the developmental timing and metabolism, energetics devoted to major life events. So bromeliads, with all life as well, starts out in sort of a growth and then maintenance of tissue phases. So we have kind of our initial parental ramet, the main rosette, growing and maintaining. Then for most bromeliads, at some point in life, we're going to switch to two main events, potentially asexual reproduction or continuation of the body through production of pups or axillary ramets, and sexual reproduction, producing the inflorescence. Whether this is once or multiple times remains to be seen. Now, a really key concept in life history studies are trade-offs. You don't get everything for free, especially if you're a bromeliad, in which things like water, light, photosynthate, and especially minerals can be very limiting. So I'm going to be looking particularly at how those interact with each other, the asexual and the sexual, under non-limiting conditions today. So sort of the first thing I've done in my attempt to make Bromeliaceae a model system for understanding life history evolution is to consider categories. And I hope this is a useful framework that you could bring to some of your research as well. And then to study variation between species. What can we measure? And I was looking for things that we can measure easily, that often don't hurt or kill the plant. Data that's easy to take with undergraduates, in particular, that's very cheap to do. This is a almost genomics-free talk. And then see what are some patterns that we might be able to generalize across Bromeliaceae. Okay, a little bit on plant development to kind of get us started here. As you probably know, meristems are critical in building plant bodies. So with rosettes, of course, right? We have a central shoot apical meristem. So the SEM image there is a Vriesea in which we've pulled all of the leaves off, and in the very center is the shoot apical meristem, which is building that main parental ramet. And I should say this sort of framework and most of the data that I'm presenting today are from a recent paper from myself and my colleagues. So in addition, as you probably know, right, every leaf has an axillary meristem. Now, in some species, at some point in develop, some of these might initiate to produce additional rosettes called axillary ramets. So when we consider the categories of life history, ask two different questions. Which meristem is producing the inflorescence? And do axillary ramets initiate? Okay, let's look at the categories. The first, and to me, what was initially the most interesting, is semilparous. So in a semilparous bromeliad, the shoot apical meristem converts to producing the inflorescence, and none of the axillary meristems initiate or are successful at making additional axillary ramets. So essentially, it puts all of its energy one time into sexual reproduction and dies. That genetic individual dies after that attempt, which is very risky. And studying in general why semilparity evolves in all life forms is a really interesting key question in evolutionary ecology. Now, semilparity has evolved repeatedly in bromeliads. Our community has not been that consistent at naming life history for species. So there are around 9 genera in which monocarpic has been identified. There's particular complexity within the large genera Puya, Tillandsia, and Alcantara. I should say that these life history traits can also vary within species. So Tillandsia utriculata, which you can see here in Costa Rica, in most of its range, it does produce axillary ramets, but in Florida, it does not. So it is behaving as a semilparous taxon as well. Semilparous bromeliads tend to be rather large compared to their close relatives, rather slow-growing, live in challenging habitats, and I think there's a lot of really important population genetic work that needs to be done in these species. They tend to have much lower genetic diversity. Very exciting to show, brand new paper, Julián Aguirre Santoro, Alejandro Zuluaga, and some other colleagues. Our big phylogenomics of Puya paper just came out this past week. Here's just a little taste of that. Julián is going to speak much more about that tomorrow. But this is part of it. We are mapping kind of as a binary life history against being in a sky island versus in interandean valleys. And semiparity is one of a suite of traits that's evolved repeatedly in sky island habitats. So come to Julián's talk tomorrow for much more exciting results from that. Okay, another life history category is single-ramet iteroparous. Now in this case, the axillary meristems are producing the inflorescences, meaning that a single rosette can produce multiple inflorescences during its life. They can as well produce axillary ramets. Again, this is something that's evolved repeatedly within bromeliads, and we are not always able to determine from a lot of our specimens or from a lot of field notes if this is indeed the case. Vriesea here in Costa Rica and elsewhere, as well as some Hectea and Dickia, this has been noted within. So I ask our community, please note the meristem origin of inflorescences in your field notes. One prediction from life history theory, I'll talk about reproductive effort towards the end of this talk. You might predict that a single-ramet iteroparous species would put less energy, biomass, minerals, etc., potentially fewer seeds, into inflorescences when it can produce multiple compared to the others, in which the shoot apical meristem produces one inflorescence. So that is an open, testable question. The vast majority of bromeliads are multi-ramet iteroparous, in which the shoot apical meristem produces the inflorescence one time, but one or more of those axillary meristems will initiate so that that genetic individual can continue on after that event. To investigate variation within this category for over 5 years now at Colorado College, we've had 3 species with large population samples growing in a common greenhouse. And we've been doing observational studies with students for many years on these 3 species. I wanted them rather small, I wanted them developing rather quickly, and I wanted them variable as well as self-compatible. So here are the 3 species that we have been growing. So we're going to look at life history traits that vary between them and look for those evidence of trade-offs between axillary ramets and the inflorescence. Now, up till now, they have gotten nothing but love, water, fertilizer, all of that, so our baseline data is under non-limiting conditions. Okay, so for the first phase of the experiment, these were all seed-grown individuals, which is really nice, because we know the origin of them. In the field, it's very hard when you see a rosette to know if it was an axillary offshoot or if it started from seed. So these are all started from seed. And we started out sacrificing 3 individuals each month of each species. Here's my students Leah and Emma taking some of that data. And we got dry biomass, leaf area, and sucrose content for those individuals. Then every 6 months, we did a survey of all remaining plants, doing longest leaf length and number of photosynthetic leaves as sort of a basic survey, and got all kinds of data. And big data sets like this on bromeliads are very rare, and they're very interesting to a colleague of mine who's a mathematical modeler, who I'll introduce you to at the end. On the QR code, there's a link, I think that works, to a video of me explaining how to take these data on plants that you could do with your students. Okay. No. Yeah. I think it's the connection at some point. Oh gosh. Awesome. Should I continue? Yes, and I'll tell you to, oh, I can do it with this. Brilliant. Thank you. Okay. Okay. So now I'm going to look this way. So here's some data on longest leaf length and number of chlorophyllous leaves. These are 3 different time slices, 3 different of those 6 months surveys that we did for the 3 species, which are different shapes. And the color changes depending on how many axillary ramets. And you can see the species basically tracking on 2 different axes. So we think that there's sort of 2 main ways that bromeliads are adding biomass. You are either a leaf lengthener or you are a leaf adder. You're not usually both, which is kind of interesting to consider. So a question I'd have for the research community is, do we think these are 2 true categories? Are these generalizable? And what other functional traits might vary within these 2 categories? Can you just progress? Yeah. Thank you. Okay, I should say that, these were data taken over many different time points, but we wanted to see, could you get some sense of which of these 2 categories a species is in with 1 time point survey in a population. The grow houses at Tropiflora in Florida very graciously allowed me and my undergrads in to look at their plants for sale and take these data. And so we have some Alcantarias, some Vrieseas, and some Neotropics, and we can kind of see, again, that species are roughly in these 2 different categories. Next slide. Okay, so this was kind of interesting too, and these are consistent things that we're seeing within a species over time. We are seeing 3 categories of the relative timing of when does the axillary meristem initiate to produce an axillary ramet, and when does the shoot apical meristem convert to producing an inflorescence. In the case of Aechmea recurvata, they're more or less at the same time. It's sort of a coincidence, all in the same span of a month or so. In strong can you go back a slide? Yep. With the Billbergia, the axillary ramets initiated way prior to flowering. Many of them were seen prior to flowering. In strong contrast to that, with the Vriesea, all of our individuals flowered and then had the axillary ramets initiate. And I've seen year after year that same sort of cycle within our populations. So again, my question to the community is, are you seeing that with your species? And what might happen once we start actually limiting the environment? Are these 2 events competing with each other or not, and what happens when we actually have some sort of environmental limitation? Next slide. Okay, so here again are our 3 species, and on the left are longest leaf length, and on the right is number of photosynthetic leaves, and we have sort of 3 categories. The green is that vegetative growth only, yellow is the inflorescence is visible, 2 first flowers opening, and then pink is flowers to fruits. And what we first of all shown, data not shown here, is that transition to axillary ramet initiation, you see no change in the parental ramet growth rate that is significant. Meaning that axillary ramets, you kind of get them for free. In strong contrast, as you'd expect, of course, is that the inflorescence does show a slowdown in that parental ramet growth rate, and it's proportional to how you are building your body. So leaf lengtheners are slowing the rate at which they lengthen their leaves. Leaf adders are slowing their rate of leaf addition. Next slide. The final phase of the experiment was a very fun one. Here's Josh Felton, one of our speakers, when he was young and excited to see flowers. And then the end of undergraduate time, very tired from massing so many dried inflorescences. So we took all of those individuals down, broke them apart into these different portions, and got dry mass as well as percent nitrogen of the tissues. And one of the metrics we were hoping to get out of that is what's called reproductive effort. Now that can be quantified in many different ways. A very simple way, right, is just the percentage of the mass devoted to reproduction versus the total. Next, please. Okay, so here again are our 3 different species, and we have the longest leaf length prior to reproductive onset, and reproductive effort, as I just showed you how that's calculated. And interestingly, Vriesea is scaling in a different way than the other two. So to put it another way, leaf lengtheners do not increase their reproductive effort at larger sizes, but leaf accumulators do. Next slide. This is real new data. This is our nitrogen data, percent nitrogen concentration for carpels, leaf, scape, and seed for our different species here, kind of massed from multiple individuals together. We haven't really analyzed it in much detail yet, but Vriesea is sort of an outlier. It has a much higher in general nitrogen, particularly in the seeds. Next, please. And so I'm very excited that the next phase of all of this is going to actually be experimental. We're going to be limiting nitrogen in our plants and looking at how this interacts with CAM photosynthesis and nutrient cycling with these colleagues. I'm going to have you skip like 3 slides since we're running out of time. One more slide. Okay, so, because I have 2 minutes left. Oh, 5, sorry. Okay, go back. I saw that too. I was like, oh God. 4, okay. Okay, cool. Okay, so why might this matter or be worth studying? Well, let's go back to semel parity for instance, because I would really like the bromeliad community to prioritize studying semel-parous bromeliads. One of the major hypotheses about why semel parity evolves, and this work has been particularly put forward in the agave community, is called the reproductive effort or Big Bang theory, which basically says that semel parity here will be an accelerating function, or reproductive success is an accelerating function of reproductive effort. And what you might see as an outcome of that is just a proportionally larger inflorescence. A lot of these semel-parous bromeliads seem to have very large inflorescences. So all our work so far has been on multi-ramet iteroparous. I'm not having a lot of success in greenhouses in Colorado in coaxing semel-parous bromeliads to flower. However, in Florida, there is a nice natural system with Tillandsia fasciculata and Tillandsia utriculata. Next slide. And the time is ripe for this sort of work because I think life history in particular for those semel-parous bromeliads is a specific conservation risk. Now specifically in Florida, there are invasive weevils predating large bromeliads. Data from colleagues is showing that utriculata is more susceptible to threats of the weevil than fasciculata. There's many reasons why that might be the case. But we posit that it's because of life history. So if a Tillandsia fasciculata had initiated some of its axillary ramets prior to predation, and they essentially were independent, not really calling on that parental ramet for resources, it might be able to bounce back in a way that utriculata simply cannot. So data that we're generating, and I'm going to show you amazing high school students that are generating data as well, are being fed to my colleague, Dr. Aaron Bodine, who's a biomathematician. And basic parameterizing data from the field and from the greenhouse are really exciting for colleagues who work in mathematics. And math students love touching plants. So here's a bunch of math students that got to go to Florida and engage in this original research. So they would want you to know that that's really exciting. Next slide. And then furthermore, something we're very excited about are these amazing high school students in Miami-Dade County, Florida, that are taking data on those 2 species to quantify reproductive effort for us right now, really expanding the reach of this research. Next slide. Okay, to summarize, big messages for the bromeliad research community. Please include consistent life history categorization when describing your species and annotating your specimens. Note the position of the inflorescence, the presence or absence of axillary ramets, and check many individuals in the field. Next slide. The life history variation in Bromeliaceae allows us to test evolutionary ecology hypotheses with simple, inexpensive data in greenhouses. And greenhouses are great, especially if you know they're started from seed. They're great for engaging early career students in science on a limited budget. Manipulative experiments and field-based studies are going to add a lot more detail and complexity to this story. Finally, next slide. The impact of life history on bromeliad conservation is important, and it requires a lot further study with all kinds of different methods. With that, I would like to thank you for your attention, my colleagues, and my former students. Thanks so much.